Welcome back. Welcome back to Wonderfest. As director of Wonderfest, it is my pleasure to introduce a Wonderfest veteran and treasure. It says that on your button somewhere. Wonderfest veteran and treasure. David Deamer, professor of chemistry at UC Santa Cruz, was a speaker at the very first Wonderfest in 1998. He made a second brilliant appearance in last year's festival where, playing devil's advocate, he convinced me that life could not have evolved here on Earth. And he goes for the three-peat here this afternoon. David has kindly agreed to discuss and to play his celebrated DNA music. Please join me in welcoming Professor David Deamer.
Well, I hurried over here from my daughter's soccer game, and I'm hurrying back after my talk to the Halloween festival, so very nice to be here for this little period of quiet time, I think. I do a lot of work with NASA funding, and this is a program called Astrobiology, and I always like to put a little plug in for that. So I'm going to start with our galaxy up there on the screen and point out that we and all the DNA in our bodies actually comes from stars. As you see here, there's 400 billion stars in the galaxy. And part of the work goes on in my lab is trying to figure out how it all began about 4 billion years ago. We know a little bit about it. We know that, for example, stars explode. And this is something that is quite a recent understanding. The past maybe 40 or 50 years, I would say, we realize this is a very important thing. And all of that dust that you see coming out from the Cat's Eye Nebula, this is a star probably about the size of our sun that blew up some, thousands to millions of years ago. All of this stuff you see here is dust, and on the dust are organic compounds, and those compounds take part in the development of solar systems such as ours. Here's a beautiful slide. Is it possible to dim the lights just a bit for this part anyway? Okay, I guess not. Okay, so here is a star-forming region in the near neighborhood. Notice all this dust from all the stars that have exploded in the past, aggregating into clouds, light years across. You can see some of these clouds here. This is where solar systems come from. And each of these stars you see whose light pressure is blowing away the dust from near those stars has the potential to become a solar system and to develop life. Now, this is the big picture, 13 billion years of time, starting with what is now the consensus view of the beginning of our universe, the Big Bang. Starting here, the first stars came along about 400 million years after the actual Big Bang because the universe had to cool down for a while. And then there could have been no life because stars had not had a chance to synthesize what we call the biogenic elements. These are carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur. And that begins in this period of time. And only after some of the stars have gone through their lifetime and undergone an explosion is there enough of that dust out in the interstellar space to start up new solar systems, and as well as our Earth. Here is the list of biogenic elements. If we could examine one of your cells and not count ions in the cell like sodium, potassium, and chloride, you would discover that these elements make up 99% of a live cell in your body and in all life on Earth. These elements are synthesized in the stellar nucleosynthesis process, and then after the star goes through its contraction and explosion, they are blown away into the surrounding universe, as I've shown you, to begin the solar systems. Now then, look at this. Here are those biogenic elements. This is a DNA molecule, and the DNA molecule has phosphorus. Oxygen is red, phosphorus yellow. Blue is nitrogen. Carbon, you can't see quite as well, is black. And hydrogen is white, making this beautiful double helical structure that we're all familiar with. So literally, we are made of stardust, as Carl Sagan used to love to say in his Cosmos programs. And this is one of the great revelations of our time, that we are really part of a much larger universe. Well, I got interested in DNA quite some time ago. My father and I, in fact, made the very first DNA model that was on the market perhaps 40 years ago now. We called ourselves Nova Research, and it's one of the great times of my young life, working with my dad, who is a hydraulic engineer, to put out a DNA model. And we sold maybe 200 or 300 of these all over the world, and that paid for the Buick that I drove to California in 1965. So I made a little bit of money out of DNA, and I still am, because my lab is interested in making what is called a $1,000 genome. Right now, there's only 2 people whose entire genome are available to us to study. One is Craig Venter, the private guy that made the genome available, about 2000, in a competition with the government work going on. And of all people, Jim Watson, who just was handed a little while ago a CD with his entire 3 billion base pairs all listed on that CD that was given to him by one of the companies doing this. That cost the company close to $10 million. NIH is giving money out now to get it down to $1,000. And my lab, with perhaps a dozen others worldwide, is trying to sequence DNA so fast and so accurately that we could give each of you your genome on a CD for about $1,000 if you had that money to spare. And by the way, I had a little bit of my DNA sequenced with my brother Richard, who's a physician down in Ventura, my brother John, an architect up in Mill Valley, and we sent it off to one of these commercial companies. So I actually know a little bit of my own genome. It's just a few hundred bases, but it was enough to tell me that, yes, I am of European extraction. This is what a human embryo looks like. Each of these cells, this is the 8th cell stage, each of these cells has a nucleus, and in each nucleus is the entire message that makes up a human being. So every one of us in this room at one time looked like this. And if by some chance you happen to fall apart, you would have become twins, because each of those cells is pluripotential. It has the ability to develop into a whole new organism, a whole new human being that would have been your twin. So one of the things that we're interested in now is to understand how that genetic information codes for the production of what we call the phenotype, what you look like after you've gone through the development process. And here's the phenotype of a single bacterial cell, and right there is the DNA in this bacterial cell. You can just kind of see it filling up that center of the bacterium. Bacteria don't have a nucleus, by the way, so the DNA is just almost naked in there. That DNA is about this long, maybe a millimeter long. Your DNA is about 3 feet long, and that's one of the differences between you and a bacterium. We have a lot more DNA, but don't get too proud of our DNA, because an earthworm has nearly as much protein genomic sequences as we do, about 20,000. We have maybe 25,000 sequences that we can turn into proteins. So we do it in some funny way, and the difference again between us and my 2 daughters, the soccer players I just mentioned, is that we have learned, we eukaryotic organisms, have learned to use our DNA more efficiently. And in fact, some of our genes we now know don't make just a single protein, but different parts of the gene and in different directions are used to make multiple proteins. So even though we have only 25,000 genes, there's probably close to 1 million different proteins that make up the human body. That's something to keep in mind as you teach. Well, let's talk about what a piece of DNA is. Proteins and DNA are called polymers. A polymer, as probably most of you know, is made up of monomers, and if we think of monomers as pearls, a polymer is a string of pearls where the pearls are attached one to the other by what we call covalent bonds. Proteins are also polymers. Again, a string of pearls, proteins and DNA and RNA. And I point out, as I realized about 20 years ago now, that a melody is also a polymer. And in my teaching, at the time I was teaching cell biology, I had the hardest time trying to get across the idea of what we meant by a sequence, and a sequence could somehow contain information. So that was really the birth of what we now call DNA music. And we were first, in fact. We had a little company. We did some cassettes that we began to sell. The company's still in existence. And if you go to the web and look up DNA music, you'll find Susan Alexander, who I'll tell you about a little bit later, who's now in charge of the company and distributing the DNA music. Well, if a melody is a sequence of bases and of notes, and DNA is a sequence of bases, maybe we can turn bases into notes. And let's take a look at what we mean by that. So here is, in a melodic sense, a set of notes. We could call them monomers. And when we string them together into a polymer, we have, in fact, something like a DNA or RNA strand, except it's a musical notation. Once again, this is what we mean by that. This is a string of letters called a sentence. And obviously, there's a whole set of monomers that make up various polymers within that, and all of that makes sense to you because you all speak English, and so you'd be able to understand the sense of that sentence. If you're a musician, and probably some of you could look at this melody and recognize it, you would recognize this as Carmen's great aria when she comes out in the opera Carmen and sings about her lost love. This is a string of bases called DNA. In the same way that the sequence of letters turns into a sentence that makes sense, and in the same way that a string of chords turns into a melody, this string of bases turns into a protein after it goes through the translation process in your body. Oops. So this caught on. Walter Cronkite sent a team, and I had the pleasure in the mid-'80s of sitting down at my little piano in my house, and a team of Cronkite's Universe filmed me doing this DNA music that you're going to hear in a little bit. And then they didn't like what I did, so they turned into a professional musician. And I had just watched this professional guy do it on the actual program, but that's okay. It caught on, as you can see in this New York Times from about 1992, I think. DNA can hum a tune, and a whole set of new products now are out. And again, just for fun, you should go to the web and see what DNA music has turned into. There are musicians taking this very seriously as an inspiration for their composition. And you can listen to some of that on the web. Okay, let's make some music. First one that we tried is called the ALU sequence. And we tried this because a friend of mine, Carl Schmidt, at the Davis campus, where I had my lab at the time, was the first to do the sequence of ALU. The ALU is just an acronym for sort of the bacterial and restriction enzymes that are used to make it. So don't worry about that. It doesn't really make sense to you. But what does make sense to you is that the ALU consensus, and here's a little piece of it, is about 10% of the DNA in your body and in mine. This is, in a sense, an infectious piece of DNA, parasitic DNA that got started in our body. We're just now attaching some function to it. It proliferated in our body starting about 4 million years ago, as I understand the bioinformatics of this, and now we all carry it. And some people call this selfish DNA or parasitic DNA or junk DNA, because this doesn't code for proteins, but it does have a very nice piece of melodic music in it. So, it's very simple. Let's make C, cytosine, into the musical note C. Let's make A, adenine, into the musical note A. Let's make G into the musical note G. This is guanine. Finally, thymine, a little bit of a problem. It doesn't have a musical note attached to it. So just for fun, we made that into an E. So I'm going to play those notes for you now. There's cytosine C. Here's adenine A. Here's guanine G. And thymine E. Now, if you're at all musical, you know that that is a C major chord, a C major sixth. Isn't that pretty? It could be an A minor seventh if we wanted to play it in a minor mode. So it's as simple as that to translate directly from the sequences of bases in DNA into a musical sequence. And if there's 3 in a row, and sometimes you might have 3 G's in a row, we just decided arbitrarily to give that 3 beats. So if we have something like this, 2, 3. You notice that some of the notes are a little bit longer, some are just a single beat, and those longer notes have either 2 or 3 bases in a row. Okay, I'm going to show you one more thing before we try some music. And that is, this is the ALU consensus as we first translated into music. And let's just go through it here. Cytosine, thymine, guanine, 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 cytosine, guanine, thymine, guanine, guanine, E, G, A, and so forth. And you just heard that, so let's just try it again. I'm the only person in the world who can play 10% of the human genome from memory, okay? Is that pretty? This comes in waltz time. I couldn't play that in 4/4 time very easily. So that's a melody. That's the very first thing that we discovered in the human genome, right out of the ALU consensus. That's 10% of the human genome. Now then, we began to use this in teaching. And the way we used it in teaching was to go to the insulin gene. Insulin, of course, is a little bit different. The insulin gene had just been sequenced when we started to do this. So this is the second thing that we did. It's a little bit different. You're going to hear how different it is. Because insulin, the insulin gene actually codes for an amino acid in the protein called insulin that we make. So 3 T's in a row is the codon for phenylalanine. GTG is valine, AAC is asparagine, CAA is glutamine, and so forth. So you really can go directly from what is a pure genetic code into a musical composition. It's going to play just a little bit of this for you, then we're going to hear it as a professional musician plays it, and I'm also going to tell you a little story about a teacher in Illinois about 10 years ago. So here it goes. So those are the first 4 bases, the first 4 amino acids in insulin as encoded in the DNA. And it kind of makes sense. It comes in what is it? Well, it's 4/4, 6/8 time. So that's kind of a jig-like rhythm if you know Irish jigs. Well, what we're going to do next is to listen to this as a musician plays it. And we're also going to watch a little piece of DNA on the screen. But I do want to tell you a story about this before I do that. And the story is that a science teacher, and many of you are science teachers I know, science teacher in Illinois decided to let her class do this. So she gave them the insulin gene and had all of them make up their own composition, just as I've done it here. I don't have to choose those notes, by the way. I could have chosen any notes that I want, because it's really quite an arbitrary choice. But she let her students do it. Students loved it. They just went to town on this. And pretty soon they had a band playing it. And it got to be so well known. She got some newspaper publicity, and she won Science Teacher of the Year in Illinois in 1992. So let that be an inspiration. I hope some of you might want to try that. Okay, let's just listen quickly to the insulin gene. And this is going to come out, I hope, through the speakers. That's my understanding. And what you want to listen for is the notes, of course. You'll recognize them now because I've played them for you. And you're going to hear 2 melodies repeating one after the other. Insulin has 2 chains, and you're going to hear both of the chains. So let's listen. That's one insulin molecule. The ribosomes in your pancreases, in the islets of Langerhans that make insulin, are making thousands of those per second. Each ribosome, at about that rate, by the way, is making one protein by adding about 1 amino acid per second, plus or minus. And you have thousands of ribosomes per cell. You have thousands of cells in your islet cells, and you're making insulin right now at that rate. So that is really how wonderful it is to understand at a molecular level what life can do. Okay, I'll just close with one other way that we can make music. This is less explored than what I showed you. It's much richer musically. So if you are interested, I can tell you how to do this. And Susan Alexander, the person I noted, wanted to do this with me. Susan was a professional musician. She said, 4 notes are not enough for me. I want a full scale of notes. So we went to the infrared spectrum of DNA and found the 4 bases. And this is the infrared spectrum of the base called adenine, which is the A that we're playing with. And this is what it looks like in the infrared. And you can see these absorption bands that are rather specific. And these absorption bands are way up in the gigahertz range, far beyond the ear being able to hear. So what we did was to bring those down in proportion, all the way down 35 octaves. And when you bring those frequencies down 35 octaves, they turn into musical notes. And here are some of those absorption frequencies in the infrared. And here's the note that comes out on the piano or the synthesizer. These are what we call a microtonal scale. So if you hear this music, and any of you could hear it if you wish, you would hear it's a little bit funny, it's a little bit off. It's not the scale that we're used to. But this is a scale of adenine. Susan Alexander composed within this scale. She put out a CD called Sequentia, which I've had the fun of listening to on the radio a couple of times, just driving along, Oh, hey, listen, there's Sequentia. So it's a nice piece of music. It's played with real musicians. She has voice, tabla, her synthesizer, violin, and cello. So it's a beautiful piece of music. I recommend it to you. And you can find Susan on the web if you wish. Okay, I'm going to finish up with just a few stories about DNA music that have come out of my experience with it. When it first came out, we began to get letters. And people were deeply interested in the possibility that somehow DNA was linked to some nervous activity or whatever. And, there's nothing mysterious. I'm not trying to mystify you, but people, of course, will take it that way. So I had a wonderful letter from a Native American woman who was a shaman, or whatever it would be called, in her tribe in Arizona. And she said that she took this tape, the DNA music tape, to her tribe, and they have a sacred part of Sedona, where these beautiful red sandstone rocks rise up out of the desert. And she played it as part of one of their ceremonies, and she swears this is true. Birds circled down and came and landed around them to listen to the music with them. That's one little letter. Another letter was very touching. It's from a woman in Washington State who had the BRCA1 gene. This is the gene that causes breast cancer if you have it. So at least a good relationship, a correlation, very strong correlation. And she wanted me to compose a bit of music in which I had corrected the wrong note in that gene. And it's only one note out of several thousand. So I took a little chunk of it and corrected the note, put it back to the normal one. She writes that she now hums that to herself after she recovers from the treatment for her breast cancer. Last little story was from a woman in Southern California. And she wrote to say that she went down to the health food store and she bought pills filled with DNA. And she took 1 or 2 of those pills every day to help her body out. And she says, by the way, I'm an exotic dancer, and here's some photographs. And yeah, her body was okay, I can tell you for a fact. So that's how some people have used the DNA music. And I'd be very happy if you pick up on it and use it in your classes. And I'll be happy to answer any questions that you have. Okay, how did I decide which note? Okay, octaves, you understand how we decide which note. We just took it right out of the letters that happen to be the right ones, abbreviations for the bases. But the octaves and the tempo and the time, 3/4, 4/4, 6/8, those are all our artistic use. So we simply impose that. But if you saw this music, a molecular biologist would be able to recognize the gene knowing the note order. So everything else, though, was arbitrary. Okay? Yes?
Could you put up the slide again on the insulin sequence? I noticed that a given, to me,
Yeah,
There's GLY down in the corner and another GLY difference.
there's redundancy in the genome. We have 64 possibilities, and some amino acids have more than one specific codon that codes for them.
Does uracil look the same one as thymine, or do you pick a different node?
Yeah, uracil replaces thymine in RNA. So there's really 5 total bases in nucleic acids. DNA has 4, and thymine is replaced by uracil in RNA. So the RNA is simply the complementary, if it's a messenger RNA anyway, it's the complementary set of notes. And it would sound the same if I played it backwards, I think. But we really haven't. We would make uracil into the note E if we wanted to play RNA. Does that answer your question? Okay. Yes.
Because of bad timing on the part of the director, I'm very sorry. If I get my hands on the guy, I'm going to wring his neck. If you would be so kind as to answer some questions, maybe audience members would be willing to go out in the hallway here. But we need to start the next program imminently. So thank you once again, Professor Deamer, very much.